My name is Wade Dizelle, and today I'll be presenting research that I conducted on the effect of a destabilizing F56H mutation on human gamma D crystalline thermostability. Cataracts are caused by the aggregation of proteins within the eye, leading to characteristic lens opacification that is a trademark of this major cause of blindness. Although aging is often cited as the major etiologic factor behind their development, cataracts can also be congenital or result from trauma, steroid use, diabetes, and even oxidative stress. Lin et al. Demonstrated the significance of cataracts in increasing incidences of falls, traffic accidents, and decreased quality of life. Due to the significance of this major public health concern, investigation and modeling of known aggregating mutations, as well as other potentially stability impacting mutations to human eye lens proteins are necessary to better understand disease progression. In keeping with this impetus to further our understanding of lens protein mutations, we produced an F56H mutant of human gamma D crystalline in E. coli cells, and we analyzed its thermostability by thermal shift assay. We selected the phenylalanine at position 56 for substitution with the histidine residue because, to our knowledge, this position has not been previously examined, despite its interesting location in the core gap region of the tertiary structure, which you can see here in Figure 1. Furthermore, histidine possesses a few interesting qualities, notably retaining aromaticity, as well as being a pretty polar molecule and an effective H-bond participant. Because of these qualities, we predicted that this mutation would decrease the thermostability of the protein, and interestingly, the results of our assay appear to support that hypothesis. So, Figure 2 provides a flowchart of our procedure. We utilized the modified quick change protocol provided by Kosinski-Collins et al. We designed two synthetic oligonucleotide flanking primers containing our two base mutation of interest into our plasmids, which are linearly amplified. A DPN1 restriction enzyme was then used to ensure that only our desired mutation-bearing plasmids remained. Figure 3 shows the results of a successful conformation shell, indicating the success of our digestion. Confirmed mutant plasmid is then transformed into nuclease-deficient E. coli cells, which are then used to produce large quantities of our plasmid, which was purified and sequenced at another facility. Our mutation-bearing plasmids were incorporated into another E. coli line, and the production of our protein was initiated through IPTG induction of the lac operon, ensuring that our protein was produced when desired and was continually produced thereafter. Cells were lysed and their contents were purified, and the presence of the mutant crystalline was detected in purified and eluted samples as shown in Figure 4. The curve of the thermal shift assay of wild type R14C, a known pathogenic aggregator, and our mutant can be seen in Figure 5. As you see in Table 1, the F56H mutant demonstrated a 1.45 degree decrease in the average melting temperature relative to wild type, whereas the R14C demonstrated a 2.88 degree decrease, suggesting that our mutation had the predicted effect. That our mutation elicited a melting point decrease, only half that of the R14C did not escape us. There are many potential explanations for this phenomenon. Whereas F56H is somewhat buried in the interior of the gap region, R14C is located at the periphery of the protein, as is shown in Figure 6. Although both residues allow for hydrogen bonding in the affected residue, the R14C is far more accessible. Indeed, Pandey et al., through Raman spectroscopy and modeling, predict that not only does the R14C cysteine participate in intermolecular disulfide cross-linking, but also extensive hydrogen bonding, all contributing heavily to the aggregation propensity of the mutation and potentially to its decreased thermostability. Furthermore, inspection of the three-dimensional structure of the wild-type crystalline suggests that the introduction of a polar H-bonding residue into the gap region, like F56H, may have limited consequence to the structure and stability of the protein as a whole, as there are a number of exposed polar residues within the vicinity that may sustain similar interactions. This could be a target for future investigation. Thus, we conclude that our F56H mutant demonstrated decreased thermostability relative to wild type. However, future studies will need to be performed to better assess the mechanism through which this is achieved. Further investigations into the stability and aggregation propensity of F56H mutant crystalline, as well as other variants such as R14C, pose the possibility of informing not only our understanding of cataractogenesis, but also the design of therapeutics. Future studies will compare the impact of UV-induced photodamage, exposure to which has been demonstrated to enhance cataractogenesis in vivo, on the in vitro thermostability and aggregation propensity of F56H mutant and wild-type human gamma D crystalline. Indeed, considering the global burden of disease, it is vital that we investigate all possible roads which may yet lead us to better understanding of, or outcome for, those with cataracts. Thank you very much.